So our leadoff hitter today, Jim Collins, got a call in 2003 that he thought was of the obscene variety. Are you Jim Collins? Are you alone? The caller asked. But the person on the other end of the line quickly informed Collins that he was the recipient of a MacArthur Genius Grant Award. And suddenly his friends all started asking him the difference between an alligator and a crocodile, the probability of life on other planets, and how much a soul weighs. I guess those are the questions that you get asked when you're dubbed a genius. So in his day job at Boston University, Collins studies systems biology, synthetic biology, and antibiotics. His technologies and patents have been licensed by more than 25 biotech, pharmaceutical, and medical device companies. One of his latest projects is featured in the October issue of The Economist, and that's to create a drink laced with bacteria that kill other cholera-causing bacteria after they've set up shop in someone's intestines. And that idea is being developed by a new Cambridge startup called SynLogic, whose funders include the Bill and Melinda Gates Foundation. So get ready to learn a little bit about paper-based synthetic biology, and please welcome Jim Collins.
So I'd like to thank Scott for the kind introduction, and even more so to thank the organizers for the invitation to come back. I was really thrilled when I got the email and replied right away to say I'd love to come talk to the audience here. And what I want to do over the next 20 minutes or so is really just share with you some new developments in synthetic biology, leading eventually to this notion of paper-based synthetic biology. So, let's go back. So this area, this oddly sounded field of synthetic biology, is a relatively new field that's bringing together engineers and molecular biologists to build biological parts, biological circuits, biological machines, and to use these devices to reprogram organisms, be they bacteria, yeast, or mammalian cells, including human cells, to endow them with novel functions, and then to use these rewired organisms for different applications. The field goes back about 14 or 15 years ago, and we participated in the early parts of this, whereas engineers, we got intrigued, what could we do in molecular biology? And as you know, engineers like to build stuff, so the kids would become engineers. In my day, it was you'd play with erector sets, so you'd go into the basement or garage, find stuff, and put it together. Nowadays, kids play with Legos. And with Tim Gardner, my star student, we sat back and said, could we do a similar approach in molecular biology? So we took inspiration from electrical engineering, electronics, where an engineer might come up with a schematic for a device, a switch, a circuit, and then model it mathematically. And we realized you could do this in molecular biology, and just as an engineer would go find the parts for that switch. So electronics, that might be a transistor or a inductor, capacitor. We realized you could do that by finding parts that would be biological in nature. It might be a gene or a bit of DNA or some other element inside cells. And we could put these together and get them to function inside living cells. And we got excited. What could you do first to start off? And what we zoomed in on is what's called a toggle switch. So these are very simple switches that are in the heart of the personal computers you carry on, the tablets, the smartphones. So these are memory elements that electrical engineers make that can be flipped, say, between an on and off state with just a transient burst of electrical signal. And really, again, there are thousands, if not really millions, that make up computer memory. So how would you do this inside a cell? And so our idea was what you could do is come up with two different genes. In this case, you have on switches. So an on switch would be a strip of DNA which would turn the gene on. If a gene is on, it's going to make a protein. So we have two genes that have on switches where they always want to be on. You then set it up so that the protein made by, say, gene A wants to bind to the on switch for gene B, shutting it off. And the protein made by gene B wants to bind to the on switch for gene A, shutting it off. So each want to be on, but each is trying to shut the other off. Right? So imagine, probably many parents in this room, imagine you're driving your car around, you got two kids in the back. They each want to talk, but they each want to shut the other up. Right? So they're trying to, and they're shutting each other. They say, you shut up. No, you shut up. Well, we designed the molecular version of this. And what you can do if you balance it right as an engineer is you can actually have it in one of two states. Either gene A is on and gene B is off, or gene B is on and gene A is off. And we showed you could do this inside bacterial cells. You could do this in yeast cells. And it becomes now a light switch for a gene. And it basically means you can endow your bacteria, your other organisms, with memory, just as you would in computers. And when we presented this work in 1999, it was at a science meeting on biocomputing. Then and still now, there is a lot of interest in could you use bacteria and other organisms to make computers? Could you use them to replace different schemes? And for many reasons, I don't think anytime soon we're going to have a computer that says E. coli inside. And the reason is as follows. As I was presenting this work to this conference, they were all excited, and the room was electrified. Oh, the Boston University engineers, they've created a toggle switch biocomputer. And, woo! And I said, okay, everybody, hold on a second. I said, these switches take hours to flip. All of the air went out of the room. And why? Well, the switches you have inside the machines we carry around take tiny, tiny fractions of seconds to switch. Imagine now you're going to search your E. coli computer on the web that takes hours for each of those switches to flip. Well, 4 days after you want to check the weather, you look back and say, boy, I didn't realize it was going to be so humid when I'm out on the UMass Peninsula. I would have dressed a little less warmly. Well, this notion of biocomputing then is wrong to think about as a computer. But if instead you think about programming, biocomputing, as creating programmable organisms, this is actually the dominant theme in synthetic biology right now. Can we reorganize these organisms, rewire them to do different functions? We, for example, with Pam Silver at Harvard, have recently created living diagnostics. So we've engineered bacteria that you can actually take, in this case we've done just mouse models, that can actually report on different things in your gut, whether you have inflammation or a possible infection. What Scott mentioned is a project that we're very excited about in my lab. We initially received support from the Gates Foundation to build synthetic probiotics. So many in the room may take probiotics, which are basically bacteria. In fact, our gut and our bodies are made up with many, many different species of bacteria. And interestingly, we have 10 to 100 times more bacterial cells in our body than we do human cells. And with each passing week, there's a new report as to how these bacterial cells are impacting health and disease. And you'll take probiotics to try to reorient different aspects of your health. We took a different approach, which is instead of taking natural bacteria as ingestible, could we rewire them to do different applications? And what we've focused on is could you engineer lactobacillus of the type you find in yogurt to basically sense and respond to different gut pathogens? So with support initially from the Gates Foundation, we engineered these to respond to cholera. And it's set up, and you see in the schematic, there's a circuit that we've endowed the switch, these cells with, such that they respond to small molecules that are made by cholera. If it senses those two small molecules, it flips on a switch that will produce an antimicrobial peptide, a small molecule that will kill cholera. So our notion here is you could use this as a way to basically either strengthen an individual in the face of a cholera outbreak or actually treat them. And we would think of basically spotting these into yogurt or to create a pill. And so in the face of a cholera outbreak, for example, in post-earthquake Haiti, a person could take this, and there's two key advantages. These are very cheap to make. And in fact, many of these small molecules can be quite toxic, but in this case, the person only would be exposed to the toxic molecule if, in fact, they were infected. Now, this at present is limited to a dish in our lab. It's not ready to be distributed. But we've now moving toward animal studies, and as Scott shared, we just launched a company with support from Atlas Ventures. In fact, Atlas was the key creator of this company, as well as support from NEA, and we just got an investment from the Gates Foundation to try to broaden the efforts to address a number of issues in global health. Well, as you hear me talk about these engineering efforts, you can envision that society gets a little concerned. My background's not in molecular biology. I was trained as a physicist, moved into medical engineering, studied whole body dynamics. And so people like you get concerned, what are these engineers doing in molecular biology? Do they really know what they're doing? Are they going to make something that's dangerous, a Franken microbe that's going to escape from their lab? My lab at BU is just around the corner from Fenway Park. Will I make something that will escape and, God forbid, eat Fenway Park? Well, as you can envision, there's a lot of concern. And public interest got hyped up by the media, in part, about 4 years ago when my colleague Craig Venter, this is not a picture of Craig, this is a magazine cover highlighting Craig's work. What Craig's team did, and it was fascinating, was they announced they had created a synthetic cell. Specifically, what they'd done is they'd taken a natural bacterium, they sequenced its genome, so its DNA, they then put it into a computer, they introduced some watermarks, they then synthesized it using a number of different companies. They took that synthesized genome and transplanted it into a related bacterium and basically rebooted it and got it to live and produce proteins. Fascinating demonstration. I thought it was a marvelous technological demo. It was not, though, that they had created life from scratch, which is how the media played it. And it garnered thousands of articles. President Obama and the Pope commented on Craig's work on the same day. Peer review I've never had, peer review I hope to never have. I studied at Holy Cross, so I went to a Jesuit school, so I like to have philosophical discussions. I like to have ethical discussions, but only to a point. And I'm an engineer, so I also like to have engineering solutions. And so with this concern that are we engineering things that could be dangerous? Are we putting things in the environment? Are we putting things in your body that could have an adverse effect that you want to get rid of? And look, that can happen, no matter how good intentioned you are. So we decided, could we take an engineering approach to this? Could we program cells so that they could count and program cells so they could commit cellular harikari after some period of time in the environment or in your body, or to be programmed to be induced to commit cellular harikari if the job is done and or if the person's having a bad reaction. And with Ari Friedland and Tim Liu, Tim's a professor at MIT, we came up with a number of circuits that actually allow living cells to count. It's captured here. Just briefly, what we did is take inspiration again from electrical engineering, where we could create a daisy chain cascade of switches. What are these? These are basically switches that are all in the off state except for the first guy. The first guy's in a prime state. It waits to see a signal that might be the change in light from a time in day or maybe a protein made by the bug. Once it sees that, it flips on, and it primes the next downstream guy, who waits to see the next signal. If it sees that, it flips the next guy down, so forth, so forth. It's as if you're going to run down a hallway, opening doors to get to the end of the hallway. Turned out this worked beautifully. We could have it on timescales of 40 minutes. We could have it on timescales of 20 hours. So you can now program cells to kill themselves, in principle, after some period of days or some time. Well, could you make kill switches? Jared Kalura actually came up with a kill switch, where in this case you can have another circuit that if it sees n number of molecules, it flips on proteins that will cause the cells to explode. And they work incredibly strong and incredibly powerful. So I have two anecdotes to share with you. When we published this work, two things happened. One is, on the heels of Craig Venture's announcements, President Obama charged his bioethics commission with their first task, which was go check out those synthetic biologists to see do those guys know what they're doing and do they need more regulation. And so they set up 3 public panels and invited many of us to speak. I went and spoke to them. And nicely, at the end of their due diligence, they said, the field is early, no need for new additional regulation. The regulations that were put in place in the 70s around genetic engineering are sufficient. But what made us proud is they actually highlighted this work, the kill switch, as a much-needed safeguard that if these guys are going to be engineering things for the environment, for their labs, to go in people's bodies, we need these sort of switches. And subsequently, the federal government supported many new programs to build on this. Second, and more interesting, when we published this 4 years ago, we received a number of calls from biotech companies. They said, Jim, we want to work with you. And I said, oh, interesting. They said, we want your kill switch. I said, why? And they said, we want to basically protect our stuff against corporate espionage. I said, oh, tell me more. Your life sounds a lot more interesting than mine. And they said, what we're doing is we're spending $500 million to convert organisms, to create organisms that could convert sunlight into fuel, for example. We're worried that the summer intern is not a summer intern. That the summer intern is, in fact, from a corporate competitor or an enemy of the state. And so we're worried that on Friday, during the volleyball game, when they run in to go to the bathroom, they have a jar, they're going to go into our big reactor with our organism, steal the secret sauce, and run out the door. I said, oh. They said, so we want you to program these guys that if they're taken out of our big reactor, they explode. I said, okay, and actually we know how to do that, and we have since done that. They said, oh, we want you to go further. I said, what? And he says, when they explode, we want you to shred their DNA. That the program says, okay, I said, I'm not sure I know how to do that. We now know how to do that, but why do you want to do that? And he says, we're worried that the bad guys have Craig Venter tied up in the back, and so when cells explode, their DNA often is still present. And so you can fish that out with techniques, so if you sequence it, synthesize it as Craig did, you have the secret sauce. And we're now developing a range of new safety switches with Synlogic and other teams to try to actually make this safe and reduce any risk. And this area of synthetic gene networks is impacting a number of different schemes, and I'm involved with another company, Sample 6, that is using synthetic biology to come up with a very rapid detection test for pathogens. And they're addressing the food industry. And this was an effort that was launched out of my lab and Tim Liu's lab at MIT, received support from local investors, Flybridge, the Kraft Group, and others, received support from the state, and actually were housed in the Innovation District for their first couple of years. And I'm very proud of their success to date. But again, as you can think, you may not want living cells in a lot of applications. And so I want to share in just the last few minutes new developments from our group where Keith Pardee, a brilliant postdoc, had the insight that maybe you don't need to have living cells. What you can do is crack open living cells and basically take their machinery and run experiments just on that outside of the cell, no living cell, just things like proteins and bits of DNA. Keith had the insight to try what happens if you take that and spot it on something just like paper or cloth. And he then freeze-dried it. So you could take these cell-free extracts, spot it on paper, freeze-dry it, store it for some period of time, and then what he found was you could rehydrate it, and this molecular machinery would then act like it's inside a cell, but it's actually just on a piece of paper. So here he's showing that he can actually flip on a fluorescing protein on these 2-millimeter paper discs. Well, this opens up a lot of possibilities. He ran a series of experiments to show, in fact, that if you freeze-dry it, you lose no activity. So this molecular machinery acts as if it had just come out of the cell, even after it's freeze-dried. And more stunningly, he took these paper discs and other freeze-dried elements, stuck them in the drawer of his desk, and every month over the course of the next year did tests on it, and there was basically no loss of activity on this, which now opens up a range of possibilities. You can store this at room temperature. So when you think about the promise of synthetic biology, most of the living cells, much of the material has to be refrigerated, which makes a big problem in low-resource areas. Well, at the Wyss Institute at Harvard, where I have a position in the lab, we took advantage of their technology development efforts where we could use just now a regular computer printer to print out arrays on just strips of paper that could have these spotted discs. We further developed simple boxes where you could, for less than $35, put in an array of these to make measurements on the output of just this paper-based synthetic biology platform. I won't go into detail of this, but Alex Green developed a molecular switch. So it's shown up in the left. These are switches that just from their structure would keep a gene in the off state. It's engineered so that it can respond to another molecule, like an RNA molecule that's produced by living cells, including those by pathogens. If it's present, it would open up that switch and produce a protein. In this case, we actually extended this so that you could actually have it make protein that you could see by eye. It could be a colorimetric output so that if the switch turned on, your paper disc would change color from yellow to purple, similar to like a pregnancy test, but now be based on synthetic gene networks. Well, with this, what the team worked on, and this is an area that is very big focus for my group, was they could actually detect antibiotic resistance genes in about 30 minutes. So as many of you may know, antibiotic resistance is a major and growing problem. Year by year, the number of resistant strains is going up. In the past, these were limited to hospitals. Worst place to be when you're sick is a hospital. Get out as fast as you can because of these nasty bugs. But now they're not limited to hospitals. They're on university campuses. They're on playing fields. They're in child care centers. They're out there. And unfortunately, the number of new antibiotics being developed is dramatically dropping. We need new ways to combat these, including early detection tests. And Keith and Alex's scheme was able to come up with super simple paper-based schemes to rapidly and readily detect multiple antibiotic resistance genes. Even more stunningly, the guys stepped up a few weeks ago and decided to see, could we address the Ebola crisis? So Keith and Alex, in under 12 hours, developed 24 sensors to detect different aspects of molecules produced by Ebola. They did it with only about $20 worth of material. So 12 hours compared to the, say, 80 days that it took to develop the antibody-based test, which itself cost thousands of dollars to develop. And it worked brilliantly. They could get very large readout in 25 to 30 minutes with these paper-based sensors. Each sensor costs anywhere from 4 cents to 65 cents to make. Incredibly cheap, can be stored at room temperature. These sensors could discriminate the Sudan and the Zaire strains. So you could use it both as a diagnostic, but also to understand where things are happening. And this is right now an academic demo, but we just published this work 5 days ago and have gotten a big interest, and we're working hard with Wyss and others now to see, can we move this out of our lab to actually have an impact. So on the diagnostics, we're very excited. Antibiotic resistance, pathogen outbreaks, including Ebola. We have developed glucose sensors, again, paper-based, incredibly cheap, room temp. We're moving also toward considering, can you use it for cancer diagnostics? Looking at molecules, not only to identify does a person have cancer, but could you identify the individuals who are more susceptible to a certain drug treatment and those who are resistant to another drug treatment. An area very close to my heart, and I know shared by the Chancellor and others here at UMass Boston, I'm very keen, can we develop this platform for educational purposes, as educational tools? Here's now a living cell-free way that you can have young people learn how to be trained in biotechnology. So whether that's in middle schools, high schools, colleges, graduate schools, around this country and even in other areas that may be resource challenged. This now would allow you to even have hobbyists play without the need for bacteria to check out and play with biotech. And final, an area I'm very excited about is portable biomolecule manufacturing. This is not limited to paper. I shared you can put it on cloth, you can put it on quartz, but you don't even have to have that substrate. You can freeze dry these as pellets. And those pellets don't need to be refrigerated. So now you can ship these around the world to make biomolecules on site, which includes oral vaccines, which includes research tools, which includes other drugs. And an area that we're actually now ramping up at the VEAS is we actually want to develop a smart Band-Aid that could go on, produce proteins when rehydrated by the wound to help the wound healing, but also serve as a sensor, to have basically embedded in those Band-Aids sensors to indicate, do you have an infection and is it a resistant infection? So with that, I just want to acknowledge the folks who actually did the work, the last part that I shared, in particular Keith Pardee and Al Green, two brilliant postdocs who took big risks and have taken on the challenge that I think they really would like to change the world, and they're the guys with the big ideas. Thank you.
Great talk, Jim. Thank you.
Thanks.
So we have time for a couple quick questions. One of them comes to us from Twitter, and I want to remind you, you can tweet your questions using the IDEAS Boston 14 hashtag, and we'll weave them in just like this one. So as I heard your talk, you're talking about a whole new world of sensors and machines that are on or in our bodies. Anna Pinkert wants to know, if we can reprogram our bodies, are we on our way to becoming cyborgs with all programmable parts? And it sounds like maybe there's a little bit of a negative tinge to that, the word cyborg.
So I think we already have cyborgs in the sense that we have, I'm sure many in this room, you have machines in your bodies. I'm sure there are a large number of defibrillators. There are, whether for your heart or for your brain, there are a large number of people who also have insulin pumps. So we are already moving toward a number of parts. I don't think we will move towards, we'll have all programmable parts, but I think we are addressing a number of issues now with engineered devices that will enable people to replace function as it's lost. And much of what I shared on the back half, I think a bigger growth area for synthetic biology will be in vitro, which is, say, outside the body for either production or diagnostic purposes.
Okay, and you and I were talking, there's a big student synthetic biology conference that's coming to Boston on Halloween, which is an interesting convergence, where they have students developing ideas for these kinds of machines and sensors. I noticed the FBI is doing an educational session at that conference. And I'm curious, you talked a little bit about some of the controls and some of the ways that government may want to get involved here. If you're in a closed-door meeting with Obama or with someone at the FBI or Department of Homeland Security, what's the one control that you would sort of say, this is something that needs to be in place in the US or around the world on this work? Or is there no control?
It's a very interesting point. So I have had the FBI many times in my office on this topic only.
With an appointment?
With an appointment, yes.
Right.
They made an appointment. And there is concern. And I think, as with all technology, there are dual-use risks. I really think anything can actually be used for problematic or negative schemes. Two comments. One is that I think there are already safeguards in place that would prevent somebody from actually sending sequences to be synthesized that would make a pathogen. So there are a number of companies that you can actually send your list of letters based on the genome you want to make, but they actually are in consortia to say, if it's coming back that this person's trying to make smallpox or Ebola or anthrax, flags go off and the guys in the black suits show up. The second, and a point that I think is relevant to the last part of my talk, and I share this with the FBI, that I understand that's their job to worry about these dual use. I'm not as worried about that. I'm not worried about the engineered efforts to make something dangerous. I'm worried about nature. And nature is much better at making stuff that's going to get us. And we see that with Ebola. right now. There's going to be another viral outbreak, I guarantee you, for the lifetimes of people in this room. We have TB killing 1.5 million people each year. These are not engineered organisms. This is nature. Nature is much cleverer than we are at making stuff that can kill us and to have it propagate. So I think we need more federal funding to do efforts to go after those natural schemes versus worrying too much about the young kids coming for Halloween.
All right.
What's your ideas?
Great. Thank you so much, Jim.
Thank you, Scott. Thank you.